Hello everyone, my name is Luca Pecoraro and I'm here to talk to you about Senecio doronicum and its two sympatric cytotypes. I'm currently doing a postdoc with Catherine Graham at the VSL. However, the project I'm presenting here today was part of my PhD that I've done at the Royal Botanic Gardens Kew in London, together with my supervisors and a lot of other people that collaborated on the project. So to start us off, Senecio doronicum is a member of the Asteraceae family or composite family. It's one of the largest plant families in the world and it's characterized by its compact inflorescence called a capitulum. Many species of Asteraceae have two different types of flowers inside their capitulum, disc florets inserted in the middle with a short corolla, and ray or leaflet florets inserted on the outside that make up the most showy part of the inflorescence. Asteraceae are a good system in which to study evolution because they are particularly prone to polyploidization, and that is the doubling of the genome in each cell's nucleus. Recent polyploids can be seen as incipient species that can differentiate over time and become completely divergent, and especially sympatric misploded population are an ideal testing ground for the effect of polyploidization. These microevolutionary processes played over long periods of time and over large scales can at least in part help to explain the diversity of angiosperms that we observe today. We studied Senecio doronicum and the Asteraceae family in the Alps, which are a great place where to study evolution because of their heterogeneity, and evolution is fueled by heterogeneity in genetic resources as well as environmental factors. The Alps have a great difference in elevation throughout the range, as well as bedrock types and microhabitats, and as well as perturbation events like the last glaciation cycles that deeply altered the floristic asset that we see today. So Senecio doronicum, as I mentioned, these are study species. This is a fairly common herbaceous plant from the montane to the alpine elevational zones, and it comes in two cytotypes, an octoploid that flowers earlier in the year and a tetraploid that is much rarer and flowers later in the year. The cross between these two ploidy levels is sterile, and the hybrids are very rare. We went around during 2016 through 2018 and sampled together with a lot of other Asteraceae, Senecio doronicum too, and we indeed confirmed that the tetraploid cytotype was very rare throughout the Alpine Arc, and in the only population that we found where there were both cytotypes occurring together was on Tête Grosse in southwest France. And this area is also known to be a hybrid zone for other plants and animal species, so it was an ideal choice for our testing of the effects of polyploidization. So this is a map of Tête Grosse in which you can see every individual plant of Senecio doronicum mapped out. You can immediately see that the octoploids are much more numerous than the tetraploids. And another thing that you can see is that the two populations, although they are in contact one with the other, they don't intermingle randomly, but there is a degree of separation between the two, spatial separation. So there is also, we can see that there is a different ecological preference of the two cytotypes, so rather maybe a competitive exclusion of one with respect to the other. As I mentioned, the two cytotypes flower at slightly different times, and this is a phenology plot of their flowering times. You can see that in the middle, between at the beginning of July, there existed almost a week in which the two cytotypes were in flower together. So although the two phenologies are largely divergent, there still exists the possibility of exchanging pollen between the two. We measured a suite of different morphological traits on Senecio doronicum, and as you can probably see from these box plots, they're not massively different between the two ploidy levels, but due to the sample size that we achieved, we were able to detect different statistically significant differences. I would particularly point your attention to the fact that the plot on the left basically tells us that octoploids make larger capitula, more showy capitula, and also taller stems. However, the tetraploids on the box plot all the way to the right, the tetraploids produce more pollen grains per floret. This points towards a different allocation of resources toward the reproductive strategies between the two ploidy levels. Still on morphology, we were interested of the relationship between the two types of florets in Senecio doronicum capitula, And as you can see on the density plot on the right of the plot, you can see that the tubular florets were more numerous for octoploids than for tetraploids. However, the number of ligulate florets remained more or less the same between the two ploidy levels. This means that the octoploids were able to fit more florets inside the same amount of ligulate florets on the outside, and this is a characteristic of the Asterix inflorescence that is known for its modularity in development and architecture. That is also thought to be one of their main characteristics for their evolutionary success and why there are so many species of Asteraceae. Overall, this is a simple PCA to see principal component analysis to see how the morphological components play together. And as you can see, the tetraploid's morphological space encompassed by the red ellipse is almost completely contained within the octoploid morphological space. This is to say that the octoploids are more variable and they contain more character combinations than the tetraploids. And the main axes of variation are, as you can see on the plot, the number of tubular florets and the height of the stem. So onto the pollination ecology part proper, we used an automated pollinator monitoring system to monitor the interaction of insects with the plants, and this system is constituted of a miniaturized computer, which can be a Raspberry Pi or an Odroid, operated by a battery, that runs a 720p autofocus webcam that observes the flower. What you end up with is these clips that are the result of a software trying to suppress the shadows, movement of the flower with the wind and other disturbances, and try to compress all the events in which there is a pollinator visiting together in one movie. The footage is also accelerated, so you can see the time frame that is skipping. This kind of monitoring footage is detailed enough that you can tell the genus and sometimes even the species of small insects, and most importantly, you can also tell the behavior of the pollinator. In this case, you can see this Eristalis going around and probing each floret, so we are confident this is a feeding visit, and this has a higher chance to transmit pollen rather than just a passing, a glancing visit. So these are some of the most common pollinators visiting Senecio doronicum. At the top you can see two hoverfly species, Eristalis tenax and Chrysotoxum intermedium, but this plant is also more rarely visited by bees and butterflies. On the plot on the left, you can see the column on the right says insect captures. These are insects that we capture flying around on Tet Cross, not necessarily visiting flowers. And there is a roughly equal amount of blue, red, and green that correspond to butterflies, bees, and flies. On the column on the left, you can see the visits of insects to the Senecio doronicum. And this column is dominated by the red and the green especially, that correspond to bees and flies. This means that basically there is a disproportionate amount of flies and bees visiting Senecio than what was available at the study site. What we also were able to see is that the duration of the visits, of the feeding visits specifically, was slightly longer for the octoploids. If you remember, the octoploids also had more tubular florets. So there is a correlation between the number of florets in an inflorescence and the time a pollinator spends on the inflorescence itself. Finally, we noticed that the proportion of feeding visits was much lower in the octoploid than on the tetraploids. You can see this on the table, in which the octoploids received together around 8% of feeding visits compared to the 34% of the tetraploids. We think that this is due to the different vegetation that was surrounding the plants. There were different numbers of floral resources available to pollinators together with Senecio doronicum, and especially on the bar plot on the left, you can see that the octoploid cytotype grew in a site where there were many more flowers from other species of plants available for pollinators. So there was a much stronger competition for the attention of the pollinators, if you will, although there was a higher diversity of species in the tetraploid site, on the bar plot on the right. Although the hourly visitation rate did not differ significantly between the two ploidy levels, neither for the feeding visits nor for the investigative visits, the two cytotypes had two distinct community compositions, so different pollinators were making the majority of the visits for the two cytotypes. On the column on the right, that is the shallower one for the octoploids, you can see that it's dominated by dark greens, and these are mostly corresponding to Syrphus. It's one genus of the hoverflies, whereas the column on the left, that is tetraploids, is dominated by the light greens. That those correspond to the Eristalis genus, another common hoverflies. And Senecio doronicum is a member of the Asteraceae family, so they are thought to have a generalist pollination strategy, more or less indiscriminately attracting all the pollinators, and as well, hoverflies are thought to be generalist pollinators, so indiscriminately visiting everything that is available. But this evidence points towards that there is some degree of preference between generalist pollinators and plants for one another. So finally, this is an MDS plot just to visualize the extent of the two communities, and the two polygons represent basically the two different pollinator communities for the tetraploids and the octoploids respectively. In red you can see the polygon for the tetraploids that contains Eristalis as a main driving factor, and the polygon in blue is for octoploids, and Syrphus is one of the main pollinators, and these polygons don't touch each other, leading us to believe that the communities are separated. To make doubly sure, we analyzed the variability of these communities with the Permanova analysis, and the two communities have a similar extent of variation, and they don't overlap also in multidimensional space, saying that basically the two communities really are different. It's not just an artifact of the different variability of the communities. So to conclude, we found that Senecio doronicum cytotypes had small differences, morphological and phenological and microhabitat preferences. And this speaks to a different resource allocation strategy in the two cytotypes. Pollinators visit more frequently the tetraploids, and this speaks to a role of flower traits and flower community contexts in the pollinator feeding choices. And finally, that the pollinator community was different between the two cytotypes, and this exacerbates the potential for reproductive isolation even between generalist plants and generalist pollinators. Well, I would like to thank you for your attention, and I'm happy to take any questions.